Good evening. Our main speaker is Dr. Soumya Swaminathan. She is the chief scientist at WHO.
It's clear that research and innovation and science has played a leading role in this pandemic, and that the public, more than ever, has been closely following the science, and this has had its advantages and it's also some downsides. Now, in this case, everything is accelerated and moving really fast, and we've seen a huge explosion of scientific publications and knowledge. A lot of it is in the pre-publication or in the preprint stage. And we see, because we review this on a daily basis, 500, sometimes up to 1,000 publications on a daily basis. So it's also very challenging to keep up with. And then today we had a fairly long and excellent session on vaccines, starting with the vaccine trial designs. And again, 15 of the 17 developers who are currently in human clinical trials shared their vaccine design protocols with us. So we were able to compare and contrast and make a presentation on the kinds of trial designs that are being considered. This is a test, I think, for all of humanity. But specifically on Remdesivir, as you know, there are many trials still ongoing on Remdesivir, including the Solidarity Trial that's looking at mortality benefit. What we know up to now is from the publications that came out of China that did not show any benefit for Remdesivir, and the publication from the NIH, which showed a reduction in hospitalization of about 30%. So patients who got Remdesivir had an average hospitalization of 11 days versus 15 days for the ones who did not receive it. We know Remdesivir is an antiviral drug, but we don't know what its efficacy is.
This press conference today is about the findings and the completion of the second large research forum that we've had for COVID-19. And those of us that are sitting up here are representing literally thousands of scientists across the world.
Because there is a very broad pipeline of vaccines. We have 4 different types of vaccines. We have virus vaccines using live attenuated virus that are commonly used also as platform for vaccines we use today. We have viral vector vaccines, which use replicating, others that use non-replicating vectors. We have modern platforms, nucleic acid vaccines, DNA and RNA vaccines, and we have protein vaccines using protein subunits and viral-like particle vaccines. As I say, an increased number of these vaccines are moving forward from phase 1 into phase 2, 17 of them. Of those who are in the clinical phase, we have one candidate vaccine from the University of Oxford, the ChAd-Ox, that you have commented a lot in the media, that is moving into phase 3. But we have other 5 vaccines that are now into phase 2.
Gosh, what have we learned? I think what we have confirmed that over time, the severity of disease seems to be standing with what we initially saw in China, which means about 20% of patients, so a subgroup of patients are developing severe disease and a smaller subgroup developing critical disease. So I think that's what we have learned and confirmed over time, and seen that same type of breakdown in different countries. We have also seen that we have learned with the recent release of the recovery trial that corticosteroids seems to be reducing mortality.
We know that personal protective equipment, in particular medical masks, respirators, eye protection, gowns, gloves, do protect health workers if appropriately used. Also, we have learned and confirmed that appropriate hand hygiene protects healthcare workers.
Almost all countries willing to share the genetic sequence data of this virus in a public database. Through GISAID, through GenBank, we have over 60,000 sequences currently shared. And this has allowed scientists to do a couple of things. In fact, the genetic sequence data was made available by Chinese scientists on January the 11th. On January the 13th, I think we had the first diagnostic assay that was published, WHO working with a lab in Germany published the first, and that allowed countries to get their diagnostics up and running. And similarly, the first vaccine candidates were also being made in January based on the sequence data, because as you know now, sequence data allows you to do a lot of things. So the RNA and DNA vaccines were all being prepared in the lab within a matter of days.
The three major trials contributing to the evidence for hydroxychloroquine, lopinavir, ritonavir, and the steroids, including dexamethasone, were the recovery trial from the UK, the active trials from the NIH, NIID, and the Solidarity Trial led by WHO. Of this, there was a discussion of the preliminary results of these trials that suggest and confirm that hydroxychloroquine unfortunately doesn't have the positive effect that we were hoping on mortality for COVID patients. There were also discussions on the potential effect of lopinavir, and the preliminary data suggests that this effect is not there, but we are looking at the analysis. And then there was the discussions on how to look into the steroids. So the recovery trial results that are publicly available were again discussed, and WHO put forward a proposition for a full systematic review of the data on remdesivir and on steroids that will be shortly shared with the community.
Can we say that the COVID-19 is losing potency as it spreads more, or what we know or what we don't know? Thank you.
So I think it's, we have to be very careful when we talk about mortality rates, and it's been suggested by scientists that really to get an accurate idea of case fatality rate, you should divide by the number of cases 2 weeks ago, not the number of cases today. There's also infection fatality rate that probably gives you a better idea of the actual mortality, that the number of people infected often is about 10 times the number of people who actually get diagnosed as cases, which is what gets counted. And so if you look at infection fatality rates, then that is a much lower number, and the figure that was presented was an average of about 0.6%, 0.6%. So that's much lower than the case fatality rates of 5, 6, 7% that we've been talking about.
I wanted to touch upon the question around the potency of the virus and the questions. We've received that question quite often, is the virus changing? And you mentioned the mutations, the changes. These are natural changes that we're seeing in an RNA virus, changes that we expect. And there's a large group of scientists that are part of this group that are looking at these viruses very, very carefully and looking at what these changes actually mean. We have not seen any indication that the changes, these mutations, these changes, confer any difference in terms of transmissibility and severity.
Is anybody in phase 3, and if I can sneak in something else in terms of vaccine, did you hear anything that relates to the question of whether or not any of these vaccines are inducing, if there's any risk of vaccine-enhanced disease associated with any of these vaccine candidates? Thank you.
Thank you, Helen. The theoretical risk of enhanced disease has been the object of extensive discussion in our expert groups. I just want to explain there are 3 levels of the evaluation. First is during the preclinical phase. So there are ongoing efforts to try to establish animal models that will allow us to evaluate enhanced disease in animal models. That has been accepted as a potential way to evaluate, but it's not sufficient. In addition to that, you have to start going through the clinical evaluation phase and evaluate it in humans. The most important part of the clinical trials to be able to document enhanced disease is to have longer-term follow-ups. And most of the trials that are being planned include 12, 15, and 2 years follow-up. And in addition, it has been clearly identified that there is the importance of post-marketing surveillance of safety as an additional element to document enhanced disease.